So I want to welcome you all to our webinar that's a unique format we haven't done before, which is we're going to interview two of our key scientists and have this be a pretty casual conversation, not as formal as some of the presentations that you've all been able to see either live or when you went onto our YouTube channel. And so I think everybody knows, but I'll say I'm Sharon Terry, the president and CEO of PXE International, and really delighted to lead this organization since its founding, since I was a co-founder for it. Almost 30 years. It will be 30 years at the end of this year, so it's quite a long time. And lots has happened. So what we want to do is to give you a kind of inside look at what research is going on. We have the pleasure of having Lili and Koen, who have both worked on PXE for quite a long time, with us to talk about that. I'll remind you that neither one are clinicians, and so while we invite your questions, clinical questions are not questions that they can entertain. Their work is really more about the mechanisms of PXE and what's happening to various body systems, and then what might mitigate, what might slow down PXE, which is, of course, a great hope of ours. And they are, in fact, really key people in how come we have any kind of treatments to test. And so I thought this would be a good format. We recently had a little blood collection happen in Philly, and they got to speak informally to some of the PXEers who came, and I realized this is really a great opportunity to hear from them and to kind of get it straight from the people who are on the ground doing the work. My other motivation, and since this is being recorded, I absolutely want to say this too, is they're both from Jefferson in Philadelphia, and Jefferson is our only PXE center, and PXE International supports that center strongly. And donations are how we do it. So you can imagine that we are collecting, $10 here and $25 there, and those donations really sustain the work of Lili and Koen, and without it, we can't continue the work there. So while it may seem sometimes to people that I constantly pitch hard, I need donations. I need donations is the reason why. And that, we know that everybody can only do according to their means, and that's totally fine. But there are 5,000 people with PXE registered to PXE International. And recently, one of our board members, Jessica Harper, who's a movie star, she calculated that if each PXEer gave $40, we could easily fund the Jefferson Center at the rate that we want to. So without further ado, what I'm going to do is I'll be the questioner here. I'll be Oprah for Lili and Koen. And I'm going to invite them to tell you different parts of the story of their work personally, as well as the PXE research story. And so they'll be talking about their own work. They may be talking about other researchers' work. We all work together, so it's really quite collegial in our big research consortium. So I'm going to start out with a simple question to each of you. And Lili, maybe you could go first and Koen second. How did you get started working on PXE, and when was that?
So I started working on PXE in December 2005. So that really goes back to 2005. I remember vividly when the first day I joined Dr. Qiaoli Li's lab here at Jefferson Dermatology. I remember that day was the day after Christmas. So that really made my memory. I went to the lab, I met Dr. Qiaoli Li, and he told me, Lili, you are going to work on PXE. So this is how I get started. Of course, I had no idea what PXE about. But then I slowly, I really began my research program starting in 2005, and up until now, I'm still working very actively on PXE, working on genetics, looking at the disease mechanism, develop therapies in animal model systems.
Great. Thanks, Lili. Koen.
Yeah, so I think a name that comes back is name of Qiaoli Li. because I say we, I started to work, I think it was in 2006, 2007, after Joni Vito gave a presentation in Innsbruck in Austria about the metabolic nature of PXE. And in those days, we were not working on ABCC6 or PXE, but what we did do is we had just developed several techniques that allowed us to screen for other proteins that very much look like ABCC6, what they are doing. And so we thought maybe we can apply these new techniques also to solve the question what ABCC6 is doing and why, if you miss it, you develop this disease. And we started all off because my supervisor of that time came up with a very interesting hypothesis during the presentation of Joni Vito, because he thought he knew what ABCC6, he didn't need any experiment. He knew what ABCC6 was doing. And although it made sense, the whole idea, it turned out that his hypothesis was wrong. So then we started to look into detail what was going on. And eventually, and I want to stress that maybe because Sharon was saying that support by PXE International is crucial, because we then got some money from PXE International to start and figure out what ABCC6 was doing. And that really was crucial for us to eventually elucidate the pathophysiology, as it's known, of this disease. So, and that is now underlying several clinical trials that have started, I think, in the last year. I don't know if they all have started. Maybe Sharon knows. So yeah, that's to emphasize that it's really crucial to donate, because without the money of PXE International, we would not have been able to do what we have been doing. And then it's very well possible that now these trials would not have started.
Great. Yeah, thank you. So it is interesting then that both of you come into the field. ABCC6 that Koen is mentioning is the gene and the protein that is awry, not working well in PXE. And in those early days, we're talking about almost 20 years ago, we didn't know what was happening in the cell, what was happening with this protein. And so the discoveries of both Koen and Lili were really big breakthroughs. And so what I think would be really interesting is for each of you to share in very simple language, because all of us need it broken down quite a lot.
That's a challenge.
It is a challenge. What work have you done over the 20 years? And if you could just sort of name the highlights, and then what are you working on right now? And Koen, we'll start with you and then go to Lili.
Okay. Yeah, so in the beginning years, we started in trying to figure out what ABCC6, so as Sharon told, the protein that's not working in PXE, what that was actually doing in the liver, because ABCC6 is only found in the liver, in the cells that form the liver. And it seems like easy to figure out what it's doing, but it took us several years actually to find out what it was doing. And after that, we have searched for proteins, because ABCC6 is a protein that's present on the plasma membrane of the liver cells, so on the out cells, the boundary of the cells, to pump out certain molecules. And what we have done afterwards is trying to find proteins that do something similar as ABCC6. Because the reasoning was that if you know what proteins are doing something similar, maybe you can find a way to stimulate these proteins. And I should add to that, the proteins that are doing something similar as ABCC6, they are not affected in people with PXE. So that should be a good target to treat PXE. And on top of that, we also now, and that's more recent work, we're looking for small molecules that you can take orally to inhibit the disease from progressing. So we are also looking into ways to stop the disease.
Great, wonderful. And Lili, what would you say your contributions along the way, and now what are you working on?
So when I joined the lab in 2005, so the Dr. Vito's lab already developed ABCC6 knockout mouse model for PXE. So the paper
And
was...
can you, sorry to interrupt, can you say what a knockout is? Because that might not be something people know.
Sure. So knockout is a deletion of the ABCC6 gene from the mouse genome. Because in patients, the gene, the ABCC6 gene or the protein does not do its job. And usually the same protein in different species does the same thing. So Dr. Vito's strategy was to remove the mouse ABCC6 gene. And when he phenotyped those animals develop calcification in the skin, the eyes, and blood vessels. So that animal serves as a very good animal model to study different aspects of the human disease. So when this model was developed, this paper was already published, we used this animal model as a preclinical platform to explore treatments. So our ultimate goal is to find a cure for patients, but we have to cure calcification in mice first. So we take those animals and we administer different molecules, compounds, drugs, inhibitors, enzymes, gene product, and then we follow up tissue calcification in those animals. So we did quite some treatment studies in the animal model system. Also, so when I joined the lab, the ABCC6 gene was already discovered. So that was 5 years before I joined the lab. So after that, we continue to do lots of genetic analysis using patient DNA samples. And we expanded the ABCC6 mutation landscape, and we identified the new mutations in ABCC6 and in other genes. And one of our large genetic study was in collaboration with PXE International, and we published that work a couple years back. So that cohort was the largest so far that I know, like 478 patients. We really expanded the mutation landscape, and we identified the mutations in not only ABCC6, but also some other calcification associated genes. So here I wanted to take the opportunity to thank all the patients and the families providing DNA samples to PXE International, which allows us to do the big genetic studies here in the lab at Jefferson.
Right. Yeah, really fabulous. Thanks for mentioning that, Lili, that those 478 who volunteered, and then we had the whatever, I don't remember if it was 9 or 11 that came back and volunteered to give blood so we could measure pyrophosphate in the blood, because that's what these guys have discovered is too low in all of you who have PXE. Those are really immense contributions. So in the same way as we thank our donors for giving us money, the donation of time and energy and literal blood and tissue has been really important for these guys to do their work. Great. So how about each of you telling us, and then I'm going to open this up to questions from you guys, because we really want you to kind of drive this with whatever you're curious about. I'm going to start with you, Lili, and ask both of you. So what are you most excited about on the horizon? Because both of you have mentioned the goal is to slow PXE down or to even eradicate it if we can. So what excites you right now in your work? So Lili, if you'd begin.
So I found this field is very exciting because there are clinical trials that is already in earlier stages of development. So those clinical trials, I have to say, are based on promising results generated from the labs. My lab, Koen's lab, and other people's PXE research work based on cell culture studies, based on animal studies. So there are, as you know, there are several clinical trials ongoing, and one of them is the recombinant ENPP1 enzyme therapy. And then the other one is a small molecule inhibitor for TNAP. I think there is a French group that is running a oral pyrophosphate trial in patients with PXE. And all those studies are based on promising results generated in the lab, and there's also a attenuated trial. So the attenuated trial, and then there was also a couple years back, there was a magnesium trial, and they were all based on promising results in the ABCC6 knockout mouse model of PXE. So that really drives me to where I am now. And as you know, there's no single therapy that is perfect. And, one single therapy is, each therapy has its own advantage and drawbacks. And I am here to continue looking into the disease mechanism. And then by looking at that, hopefully we can identify like more effective and more effective and safe treatments for the disease.
Fabulous. Thank you. Koen, what are you most excited about?
Well, I am excited about all the things that Lili just mentioned. And most of these therapies are aiming to increase plasma pyrophosphate levels. So the molecule that's low in plasma of people with PXE. And on top of that, personally, what I'm very much interested in is to figure out, but we have just started that project basically, is why is it that there's such a dramatic difference in the way the disease presents itself between individuals, even between individuals from the same disease. And why do I think that's very interesting and exciting? That is because it means that there are possibly maybe small lifestyle changes or maybe behavioral changes that can affect the disease from progressing. And if we can figure out what is responsible for these large dramatic differences, then we might just come up with some kind of advice for PXE patients that might reduce their disease from progressing. So that's what I find very interesting myself, and that had potentially also has implications for patients.
Terrific. Each of them had mentioned, for example, small molecules as a way to treat PXE perhaps, and you're all actually very familiar with small molecules. Aspirin is a small molecule that is interrupting a pain cycle or an inflammation cycle, that sort of thing. And the hope is to find either that or, as Lili also mentioned, enzyme replacement therapy, which is used in some diseases to replace an enzyme that might be missing. And so to have the body do its progression more typically. We all know, as Koen just mentioned, PXE is highly variable. No two people have the same experience, even siblings don't. And so lifestyle is important. And also finding different therapies, as Lili mentioned, might be very important, that it isn't just going to be one. The same way as you might take ibuprofen, and I hope you don't, because with PXE that actually can cause bleeding, but the average person might take ibuprofen and it works for pain, and someone else might swear by Tylenol or paramedicine. Those are different reactions the body has, both because of genetics and other things that the body has experienced over time. So I want to open up the floor for questions. You can either unmute and ask them. You can put them in the chat, whatever works best for you. If you want to turn on your camera when you're asking, that would be great. If not, that's okay. Daniel, I see you unmuted.
Hi. Yes, I'm a physician geriatrician here in New York. I teach for Presbyterian, but my wife has been afflicted, as well as her sister. My wife was diagnosed about 51 years ago at Bellevue, and she's been a patient, Mark Lebwall, for many, many years. A lot of her tissue from skin surgeries, went towards, the diagnosis and actually genetics of it. But it's been a tough wall. Again, 50 some odd years of this disease has not been an easy task. And being an internist, I'm always looking for something coming around the bend. The thing with adidronate, I'm a geriatrician and I use a lot of bisphosphonates and other drugs for osteoporosis. And my wife has osteopenia at this point, and I would love to get her on adidronate, but I know it's almost impossible to get, outside of the United States. Is there any suggestions with that? We have tried so many things. Years ago, we saw the wonderful Dr. Wito, down at in Jefferson, and everybody tried to help, but there really was nothing. And only now is there starting to be little dribs and drabs of things coming through. She couldn't tolerate magnesium, so I'm looking at something that might actually help another issue she has, which is the osteopenia, which eventually is going to go towards osteoporosis in her case because of her concern about calcium ingestion.
Yeah, good questions. Daniel, one thing I'll say, and then both of our speakers can jump in. Yes, we're right that this one bisphosphonate, etidronate, is not available in the U.S. Because it's not really available, and it's been able to be part of this trial in the Netherlands, and they're getting reasonably good results. One of the super hard things with PXE that we haven't mentioned is something called a biomarker. And PXE progresses so slowly that you need 20 years before you know whether something works or not. So while the etidronate trials, I think 5 years in, and they're seeing a few different changes that seem good, we still don't know. And as you know, Daniel, there are side effects of etidronate that we might need to be concerned about because people would need to take it a very long time. One is necrosis of the jaw. But it does seem to be doing something significant. Another question we have is, do other bisphosphonates work as well? So, if someone is taking a bisphosphonate that is typically prescribed now, would that work as well? And we haven't had those experiments happen as these guys talked about in animal models so much yet. But let me turn it over to our speakers and see if either of you have any further comments on the question.
Well, yeah, I know that in the Netherlands, it was when they wanted to start this trial, they contacted us, what would be the best bisphosphonate to use. And what they did is they had it made, I think, in Greece, because it was in those days it was also not available in Europe, or at least it was very difficult to get. So they had it specifically synthesized in Greece for this study, because the problem is with, if you want to treat osteoporosis, then there are better options to treat the disease. So that's why etidronate itself is no longer used to treat osteoporosis. But I would think for PXE, it's the best bisphosphonate to use, actually. And I am in contact now with a non-profit organization that's looking into marketing or into getting etidronate on the market for a treatment of PXE in Europe, I think. So I can ask what their plans are with respect to also bringing this to the US. Because I agree with Sharon, the results are promising. And why does etidronate work so well? That is because it looks very much like the molecule pyrophosphate that's low in PXE patients.
Yeah. And another angle we've been working, Danielle, is also compassionate use. It's a little different because this drug is not on the market the same way another one is. So there isn't a big company to go to. The trialists they've made the drug themselves. So through this contact. So yes, that I think is an important thing for us to follow up on.
So I wanted to add on, which bisphosphonate would be helpful in the context of PXE. So when we did the early on mouse study, we tested 2 different bisphosphonates, etidronate and alendronate. So I agree with Sharon and Koen that etidronate is a very weak osteoporosis drug. Because it was the first generation, and its anti-osteoclast activity was rather small. There are more potent bisphosphonates for treating osteoporosis. But when we tested the 2 bisphosphonates in our animal model, we were particularly looking at soft tissue calcification. So instead of the bone was not our major focus at that time. And what we found was etidronate was more potent than alendronate in preventing tissue calcification. So that was a little story generated in the lab even before the clinical trial in the Netherlands.
Yep. Yeah. Again, a good line between some experiments with animal models all the way out to humans, which we keep pushing. And so supporting these guys' work has been really critical to that. Okay, in terms of a queue of questions, Colleen, we're going to take your question offline because it's about you personally. Leo, you're asking what kind of lifestyle advice does Koen know will work?
Yes.
So let me start with disclaimer. I'm not sure that which lifestyle changes will work, but what I would do if I had PXE, I would make sure that I get enough magnesium, because although the clinical trial did not show statistical significant difference, but that's probably due to the fact that it was underpowered. So I would definitely make sure that you take enough magnesium, and maybe also make sure that you eat enough green leafy vegetables, because that's full of vitamin K, and you don't want to have low vitamin K because that can make disease worse. And something that we have been looking or trying to look into is also if exercise might work. And there are some indications that, let's say, exercising increases levels of pyrophosphate in your blood. But we have not been able to show that increase in plasma pyrophosphate also, at least in mice and in rats, that also reduces the disease from progressing. But still, I would still feel that exercising might also be beneficial to PXE patients. So to wrap up, enough magnesium, enough leafy vegetables to increase vitamin K, and exercise.
Right. And it is also well known that because so much of PXE mimics other diseases like vascular diseases and eye health and other things, that the things we know in general about the general population are very important. So Koen's points are spot on. Exercising, keeping your body weight at a reasonable level, making sure you eat well, all those things help everybody with vascular health and so on. And the other thing I think that sometimes gets conflated or meshed together in a way that's not helpful is often people think, oh, I'm a 68-year-old male and I have just learned that I have plaques in my arteries, and so it's PXE. It could be PXE, but 68-year-old males, especially in the Western world where we have McDonald's and high-fat diets and we are sitting around watching TV, are very common. I read a statistic yesterday because I was doing some research for somebody that in 98% of men over 70 years old have plaques in their abdominal arterial walls. So we all have to be careful that we're not conflating common conditions with PXE and that these things interact and we should be as healthy as possible. Okay, so another question for you guys is how is the liver actually affected by PXE? And are there any trials in Europe for patients over 80, or is it too late? I'll answer the second part. It isn't maybe too late because 80 is the new young 40, right? Right, Hope? But most trials are highly regulated by something called the European Medicines Agency, or the FDA in the US. And those high regulations usually mean no children and no old people. And we could all decide what we mean by old people. But in most of the trials, we won't be accepting people over. We pushed it on the recent trial for the TNAP inhibitor, and we went to 75 years old. So the trial may not be including you, but the drug that comes out would be one that could be prescribed for you. Yes. So how is the liver actually affected in PXE, since you keep mentioning the liver?
So I will start, and then Koen, you can tell me. So liver is very important for PXE because the ABCC6 gene is predominantly expressed in the liver. So the liver, so there are several different cell types in the liver, and hepatocytes are the majority. I don't know the percentage. It must be more than 95% cells are hepatocytes in the liver. And ABCC6 is expressed on the basolateral side of hepatocytes facing the blood circulation. And ABCC6, as Koen said, ABCC6 is a pump. It pumps out some molecules from the liver to the blood circulation. And if ABCC6 does not do its job, then the concentration of that molecule will be low. So now we know the connection between ABCC6 and the PXE is through ATP and pyrophosphate. So that was a seminal discovery of Koen. So Koen, you wanted to talk about the ABCC6, ATP, and pyrophosphate.
Yeah, but I want to stress that apart from ABCC6, liver function is normal in PXE. So you shouldn't worry about liver disease, basically. That's, I think, important to mention. So apart from ABCC6 not working properly, all other functions are normal. So, and yes, that's the way ABCC6 is working. It pumps out the molecule known as ATP into the blood. And then you need another protein, another enzyme to convert the ATP into pyrophosphate, this molecule that inhibits calcification, and another molecule, AMP. And the pyrophosphate is then distributed throughout the body. So it goes via the bloodstream to all different parts of the body where it prevents the precipitation of calcium and phosphate. And that's what we know as calcification.
Right. And the body normally calcifies or wants to calcify, both with healing wounds and with aging and so on. It's a repair system that's good and useful, but it is too much in the peripheral areas because this pyrophosphate's not reaching in great enough volumes in people with PXE. So you might want to say, well, then why don't I just eat pyrophosphate? that would just make sense. So what would you say about eating pyrophosphate? Since Koen, I sat in your lab however many years ago when you made this discovery and said, why can't we just feed people pyrophosphate?
Yeah, so in those days, when we just discovered that pyrophosphate was low in blood of PXCers, we thought, no, giving pyrophosphate orally, so as a pill, it's not a good idea, because what you have in your gut is enzymes that degrade the pyrophosphate. So they break it down into inorganic phosphate. So it's maybe a little bit complicated, but pyrophosphate is two phosphate groups joined together. And you have enzymes in your gut that just break up the pyrophosphate. And then you end up with phosphate. And that might be actually part of the problem in PXC, because it precipitates together with calcium to form calcium phosphate deposits in your soft peripheral tissues. So initially we thought, no, it's not a good idea to take pyrophosphate as a pill because it's degraded. So you won't get any pyrophosphate in your blood. But then one of our colleagues in Budapest, András Faradi, he just thought, I'm going to give a pyrophosphate to my PXC mice anyway. So that's what he did. And then he came to us and he said, look, I'm just giving these animals pyrophosphate via drinking water, and the disease stops from progressing. And we were actually quite flabbergasted and also a little bit disappointed that we didn't think of this. But so that's also why now people in France are doing a test with oral pyrophosphate. On the other hand, we have also done similar experiments, and we needed really a lot of pyrophosphate to come to the same result. That would be the same amount if what we give to these ABCC6 mice would be in grams. Can I use grams here?
Yes.
It's close to 140 grams of, so that's a quarter pound or something like that, quarter pound of pyrophosphate, which is an awful lot. So no human being would take 140 grams of pyrophosphate a day. Yeah, so I'm still a little bit doubtful whether that would work for all PXC patients.
Great, thanks.
And you found something similar, right, Lili? You also had difficulty. You also needed a lot, or with the concentrations or the amounts used by András, you did not find prevention of the disease from progressing.
That's true. We need a lot more pyrophosphate to a different magnitude to see some effect. But then there are the animal study. But then for the human subjects, we collaborated with András Faradi and Sharon Terry and PXC International. So we had, if I remember, we had 11 PXC patients who joined our small trial in Washington, D.C. And we really saw increase of pyrophosphate in blood, just like 10 to 15 minutes after ingestion of the pyrophosphate. However, pyrophosphate in blood quickly declines. So the half-life of pyrophosphate is very short, like 30 to 45 minutes. So that raises the question, how frequent you have to take pyrophosphate to see some benefit.
Great, thanks, Lili. So, Koen, I remember a significant moment, and Daniel put a question in the chat to this question as well, that we were at that Innsbruck meeting, and your colleague Pete Borst, who's acclaimed and famous in these transporters, jumps up, and this is a meeting, by the way, that PXC International supported and continues to support. I'll give the keynote next March in Innsbruck at this meeting, the ABC Transporter meeting. Pete leapt up and said, I know the answer, as you described, and the answer was vitamin K. And we just spent the rest of the meeting all so happy he had found the answer. And so to Daniel's question, should we just be administering vitamin K to people with PXE? What is your thought?
I think if you just have healthy diet, I would, but that's with many supplements, right? If you just eat if you have the healthy diet, you don't need all these supplements. But if you think you might be low, if you don't have a lot of green leafy vegetables, then it might help, because you don't want to have too low vitamin K, because vitamin K also helps to activate certain other proteins in this case that also prevent calcification. So it's not only pyrophosphate that prevents soft tissue calcification. There are other factors as well. And you should be sure that these are at a good level to prevent the disease from progressing. So I would think it's good to have enough vitamin K in your system to prevent a shortage of this compound, which might make the disease progress.
Great, thank you. Question from Colleen, what helps pyrophosphate to effectively metabolize? She's asking, would adding hydrogen help it metabolize?
Hydrogen. I don't think that hydrogen would help. I don't know how you can take hydrogen.
Right, and whether you could
Because
recompound.
it's a gas. Hydrogen is a gas, right? Or maybe you mean a different compound? Can that be?
She's saying.
Oh, I see it in the chat.
Yeah,
Yeah.
good.
So no, I don't think that would help. No. So what you would like to do is not to metabolize pyrophosphate, because you want to limit degradation of pyrophosphate. And that's exactly what one pharmaceutical company is looking into. They are targeting tissue non-specific alkaline phosphatase, or TNAP. And they want to inhibit this enzyme because that breaks down pyrophosphate. And if there's less degradation of pyrophosphate, then levels should go up, and maybe to levels that are high enough to prevent the disease from progressing.
Yeah, it's significant to note that carriers like myself, so if you have PXE, all your parents are carriers, don't have any effects of any tissue calcification, and we're only half doses, right? Because I have only one gene that's working well and the other gene is not, and so I have a half dose perhaps of pyrophosphate myself, and none of us have symptoms, which is true in recessive diseases in general, with some exceptions. So the point is, if we can get pyrophosphate to get up into any kind of normal, even low normal range, then we should be quite satisfied that over time, the disease would slow down. Great. Other questions?
And to add to that, we also tested in carrier PXE mice, basically, and we didn't see any calcification there either. And they were really old.
For mice.
For mice, yeah.
Old mice.
Old mice, yeah.
When we had our first knockout mouse made, and again, that's a mouse that they've removed the ABCC6 gene, my kids were still pretty young because this is more than 20 years ago, and they wanted to bring all the PXE mice to our house since they should have also a support group. But we did not.
Yeah.
What other questions do you have?
Do you think we're any closer to getting ability to attitrinate? I've used a lot of bisphosphonates for a long time. I have less fear than most people do. I think most dentists are the ones who freak out over it. However, the rare cases I've seen osteonecrosis of the jaw was always someone who had head and neck radiation at one point in their life and really had bad teeth. As far as the side effects of all bisphosphonates, just that we have better things. We have denosumab, which, for osteoporosis is a charm. It works beautifully. We don't even need the bisphosphonates as far as I'm concerned. Only the insurance carriers are the ones that want to use them because they don't want to pay for, Prolia. But I'm just curious how close, if at all, we are. My wife's biggest problem has been the calcifications on the skin that are making her life very uncomfortable in spite of multiple plastic surgeries over the years.
Yeah, so I think I know that they're working on that in Europe, but I don't know if they also, if that's successful, if they want to bring that to the US. The problem is that there's no patent on it.
There's not enough need for
There's
it.
no
either.
incentive
Yeah.
for companies to, especially with a limited number of patients, to bring this to the US, because there's little money to be made out of it. And that's a problem. But I don't know, maybe if it's successful, I don't know, maybe non-profits can step in. I don't know how this works because, you know,
Yep.
I'm just a vet.
Yeah.
Maybe Sharon can comment on that. If it's possible that if successful in Europe, if they can bring it to the US as well.
Yeah, so that's our plan is to we know you all may know I also wear another hat, which is I'm the CEO of Genetic Alliance, which is an alliance of 10,000 rare diseases, and we know in other diseases this has been solved by if we can get proof that it works. And Koen made a good point. The EMA, the European Medicines Agency, may approve this drug. The FDA may not approve the drug. This happens all the time. In fact, there was just a big study in the US about how do we get these two to work together better. But if we did, then PXE International would step in and see, is there a way that we could create a manufacturing arm of PXE International and manufacture the drug? We're doing that, in fact, with pyrophosphate. The patent on pyrophosphate for the US is now owned by PXE International in case that drug is used as a drug is useful. And we would be the ones that would then gear up and manufacture it. All of these things cost millions of dollars. And Koen's point about there's no market is a really big one, because what happens are these drug companies spend a lot of money. They actually do. I mean, we all complain that all these drugs cost too much, and Pfizer and Roche and Amgen and whoever else are just gouging us, and they probably are. And it costs, the average is $1 billion to bring a drug to market. The failure rate's 95%, and it takes 20 years. So it's disastrous. It's worse than being a weatherman who can't predict the weather 50% of the time. This is even more dismal. So we're excited when we get close to, for example, the TNAP inhibitor data looks great out of the phase 2 clinical trial. And then is it going to be cost effective when maybe you'll have 3,000 people in the world taking the drug to actually manufacture the drug? So we are looking for ways that we would be able to subsidize this so that it doesn't cost a lot, and that we could also do this simply.
Yeah, and the market will be big if there are data showing ethidronate, also reduces calcification in other clinical conditions affected by calcification. Because, as Sherman said, in elderly people, more than 90% of people have their blood vessel calcified. So if there are such data, even if in animal models, I think that will bring ethidronate closer on the market.
Yep. And that's a great point. And the other thing that we've been working on is when we've sat down with the FDA and the EMA, they say, well, I don't think the burden of disease is very high in PXE, where they look for diseases where the burden is great. So they look at things, where people die when they're 6 years old, and they say, oh, the burden's great, that's clear. And for PXE, they say, oh, people have eye injections now. They don't need anything else. The skin is cosmetic. We don't care about the skin. We've recently seen a correlation where the light-dark adaptation in people with PXE's eyes is slow. And so you probably all have experienced this, and we all do as we age, step outside in the dark, and it takes a while for your eyes to adjust. FDA folks actually said to me, so what? The mom runs out to the car, 40 years old, picking up her kids from soccer, and she can't see for 10 minutes. Well, so what? I mean, that is a huge burden. So what we've done is we've surveyed you guys, and I hope that some of you have participated and done a quality of life burden study the way the FDA and EMA want it. And we're in the process of analyzing that data right now. And the data shows burden of disease is high for skin, for eyes, for arteries. It isn't that this is a nothing disease and we don't have to worry. So part of what we have to prove is that there's a burden here that FDA does have to go out on a limb a little and approve drugs that might not be slam dunks the way, we've seen other drugs work, which again is hard because PXE progresses so slowly. There's not a good biomarker yet. We're hoping this light dark adaptation is actually a good one because it can be changed quite quickly.
Would it help, for instance, because I can see that if a manufacturer makes the etidronate, it will not be extremely expensive, I would think. Would that help making it easier to get onto the US market as well?
Yeah, I think so. But again, Kunu will have to be probably a nonprofit. So PXE, or I'm the president of a nonprofit in Geneva called Espérer. And Espérer's goal and mission is to take things that nobody else wants to pay attention to and push them into the market. And the model's going to have to be different. The drug companies have always looked for either I can make an arthritis drug and sell it to hundreds of thousands of people for pretty cheap, or I can make a new drug for a disease called Hutchinson-Gilford progeria, and I can charge $1 million a year for it, and the insurance companies will pay.
Yeah.
The problem with those models are, number one, the blockbuster model of lots of people doesn't apply here. And the second one, if the drug comes on the market at $1 million a year, sure, a bunch of insurance companies will pay. But as we get more, this past year, 280 rare diseases got approvals for rare disease drugs. If the insurance companies are even paying out 50 of these or 100 of these at $1 million a year, they're going to go bankrupt. That's just the way that the economy would work. In fact, someone said if we got 500 rare disease drugs on the market next year, it would actually break the bank of the GDP for the United States because of the amount of money that would have to go to the drug companies for the drugs. So we need a new model, basically, of how to develop these drugs.
Yeah, I agree. Yeah. Because that's also probably one of the problems with the EMPP1 enzyme therapy.
Yeah.
Yep. Yeah. Yeah. I think that's going to be hard. And the other thing with any therapy, we're never going to reverse PXE. There's not any work being done except for a macrophage thing for the skin. But the skin, for example, turns over, becomes new cells so slowly that someone is not going to have a regeneration of skin cells at the rate they would need to get rid of the skin lesions. Same thing with the Brooks membrane, the cracks in the eye. It will be much better to be able to treat people when they're 20 and all their lives so that we're not getting the manifestation, the symptom yet. So we'll need therapies that can be delivered a long time and give it to much younger people, which is why we keep trying to encourage the parents of the very few pediatric patients we have. We have at any one time maybe 40 kids in the world because PXE is diagnosed when you're in your 20s or 30s. Anyway, we keep encouraging the families. This is really for your kids. It's really going to make a difference. It certainly will also make a difference for all of you with regard to vascular health and probably this eye health issue, especially around dark light adaptation, but probably not for things like skin and for already cracks that exist in the membranes. I mean, that's definitely stuff that people are working on because there's problems in both of those areas, but it's not as easy as slowing the disease down. Reversing the disease would be very, very hard. Other questions?
As far as the etidronate, it was something that was used. I'm in medicine a long time, and that was out a long time ago. So obviously it was kicked around. So somebody was manufacturing it and probably has it in their back shelf somewhere. That's why it doesn't have to be reinvented like the wheel. It's there. So it's...
Yep. And we are talking to them and to this company in Europe that is considering it. There's another company in Budapest that's considering it. So we're doing negotiations now with what can be done. Also, how much the Netherlands group patents what they find, because they don't patent etidronate because they can't, but they patent it for a purpose. And then how will they lock that up? PXE International has patented everything we possibly could. with the goal of making it open and free, just like when we discovered the gene and when we created the genetic testing. But when we can't get in on the ground floor, and it's also why we're happy now to have the patent for PPI, but when we aren't the ones on the ground floor, we're going to have to negotiate with the people who do hold the patent, and that's going to be hard because they're all looking for a killing using it for scleroderma or other calcification conditions. So we are pressing hard. And if you have any ideas, Daniel, we're always happy to have soldiers in our war
Thank
here,
you.
because that's what we continue to do. Yeah. Okay. Well, Lili and Koen, this is where I start to get teary and cry. Your work over all these years is really what has sustained this field. If we look at the threads, as you've said, that are in clinical trials and so on, it's because it came out of work that you've done. And so we really, really appreciate that and want to continue to support you in every way we can so that you stay working hard for so many people with PXE.
Yeah.
Thank
I
you,
just want to
Jerry.
mention that I just learned, and that's a little bit sad, that one of the people that also initially identified found that ABCC6 was involved, or lesions in ABCC6 are responsible for PXE, just died. It's Arthur Bergen.
Oh, really?
Yeah, he died last week.
I think he was not much older than me. Wow.
No,
Wow.
suddenly.
Oh,
So
boy,
he was
yeah.
one of the people that in the beginning did a lot of work on PXE, and he was also quite important in getting the field started.
Yes.
I
Yeah.
think.
Yep.
So
An ophthalmologist in the Netherlands. Yeah. Really incredible.
Yeah.
Oh, well, and we know, we should also not close this meeting without saying our dear Yoni Vito, who was the boss of these guys and the guy who took us seriously when we came to meet him 30 years ago, suddenly died a couple years ago as well. And we're very happy that these two brilliant scientists can continue to work and have continued to work, having been mentored by him, because Yoni's contribution is probably the most immense in the field. So grateful that we can continue with Jefferson.
Thank you so much, Sharon, for your support over
thank
these
you.
many years.
Yes. Well, here we
We
go.
appreciate it a lot.
Good. Well, we have a lot more to do, so we'll all roll up our sleeves and get going. All right. Thank you all.
Wonderful to have met you all.
Yes, you too. Thank you.
Likewise.
Have a good day, everybody.
Thank you.
Bye. Well, that was a really fun interview to do. We really want to continue to support these scientists. What they're doing is invaluable for us as people who are concerned with or living with PXE. Here to talk to you about how you can contribute is Jessica Harper. She's a famous actor, and she's our board member, and she herself has PXE. Listen to her tell you about that and the fact that she's going to match your donation. Thanks.
Hi, everybody. My name is Jessica Harper, and I'm an actor. You may know me from movies like Minority Report or My Favorite Year, or currently on Paramount Plus in a movie called Memory with Jessica Chastain. I started my career back in 1969 in the Broadway musical Hair. But perhaps a more significant year for me back then was 1960, when at age 11, I was diagnosed with PXE. I've worked on the board at PXE International for 24 years now. PXE International works so hard for all of us. They work 70 to 80 hours a week. Our CEO, Sharon Terry, has never taken a salary, and we have scientists in labs all over the world looking for a cure for our disease, and they are so close now. But they need more funding, and that's why I'm reaching out to you today. I understand that if each of us in the PXE community gave $40 to PXE International, we could cover all our expenses for a full year, and that's pretty amazing. So I am asking you today, if you can, please scroll down on this page and donate $40. And, by the way, if you do, I will match you for every dollar you donate. So thank you in advance for supporting PXE International, and thank you for your time.